Hello everybody, I thought I give you a quick feedback on the questions that I set for the Molecules of Life module in the most recent multiple choice quiz. And we started with the question, what is the chemical mechanism by which cells make amino acid polymers, so peptides, from monomers? And we've got a choice here. So if we just quickly recall what this mechanism is. So we've got an amino acid here and a second amino acid here. And we know that the acid function and the amino function here react with each other and form this peptide bond here. This one here, and at the same time we release water. So here's the water molecule that we release, and therefore it would be a dehydration or condensation reaction. So answers E would be correct for this question. Now, in the next question, why is RNA usually less stable than DNA? And that has something to do with the number of with the position of the 2' OH group. So in RNA, we've got an OH group located here, whereas in DNA, we just have an H. That's why the deoxy stands for. So in RNA, we've got an OH group here, which can then attack basically this phosphodiester bond here and lead to a breakage of the strand. So the reason why this RNA molecule is usually less stable is because the RNA contains an OH group in the C2 position of the ribose molecules. So answer C would be correct. In question 3, it is asked that if you've got 666 base pairs, and what is the maximum length of a protein that can be encoded by that sequence? We know that a codon exists, is made up of 3 bases, and each codon basically can stand for an amino acid. Therefore, all we need to do is we take the number of base pairs divided by 3, and we get 222 amino acids that can be potentially encoded by this DNA strand. So answer A would have been correct. The backbone of a protein consists of, so if we're looking at a protein, we have usually a carboxy group, we've got an amino group, and we've got the C-alpha atom. And then we would have the next carboxy group, amino group, and C-alpha. So we have a carboxy group, an amino group, and an alpha carbon atom, so that would be A here. Of course, there's no phosphate group, there are no side chains involved, and amino groups and different side chains just simply doesn't make sense. And in question or answer E, a carboxy group and an amino group, yes, but we also have the C-alpha. So in this case, it would be answer A that is correct. Which of the following is least likely to result in protein denaturation? Well, we said that changing the pH can have dramatic effects on the charges of a protein, which then can destroy the structure of the protein. When we boil a protein, it might disrupt the hydrogen interactions and weak interactions. So that will be certainly the death of a protein. Exposure to detergents like SDS, sodium dodecyl sulfate, can destroy a protein. But adding salt to the protein is something that most proteins will certainly survive. So this is the least likely to have an impact on the structure of the protein. So salt will most likely not lead to a denaturation, and therefore this would be the correct answer. In the alpha helix of a protein, the R groups of the amino acid residues, they are, we looked at the structure of that, and we said the R groups are sticking out to each side of the helix. So they don't alternate between inside and outside. So that's certainly not correct. They are found on the outside of the helix spiral. Yes, that is certainly correct. It has nothing to do with formation of whether the helix goes like that or like that. So the R groups have no impact on the handedness. And of course, the R groups do not form the hydrogen bonds in the helix. The hydrogen bonds are kept to, or the helix is kept together by the hydrogen bonds in the backbone. That is why helix and also a beta sheet are secondary structure elements, because of the hydrogen bonds in the backbone of the protein. And of course, the R groups are not in the interior, so the correct answer here would be B, are found on the outside of the helix spiral. So these were the 6 questions that I have set. I don't think they were too challenging, and I hope you got them all right. So thank you very much for watching, and have a good break.